Well, thank you for agreeing to do this interview with us, first of all, and taking time out of your, I'm sure it's a really busy schedule. We just kind of wanted to get to know you. What first kind of drew you to this field of study?
Okay. So as an undergraduate, I went to a small liberal arts college in northern Maryland. It's now called McDaniel College. And I really took advantage of the liberal arts environment. So I studied a lot of different things. I did a lot of music. I ran track and cross country. I had a really great time. And college went by in a flash. So going into my senior year, I realized I hadn't actually picked or declared a major, which they were very tolerant of in liberal arts settings. So that summer, after my junior year, I'd been playing in a rock band at the time, and we our summer job was touring various Ocean City, Maryland, and Washington, D.C., and Baltimore. And I was so exhausted at the end of the summer that any sort of music, anything extra, I just wanted to kind of go into a focused, focused feeling going into my third year. And at that point I realized, looking at the courses I'd taken, I sort of said, oh, I seem to have been really interested in a lot of psychology and related courses. So it wasn't difficult for me to complete that major. So, and I also started reading in the field really heavily and preparing for the GREs and that kind of thing. So it was really only my last year in college that I kind of clamped down into psychology. Because it was a liberal arts college, I hadn't done much research or anything. Most of the faculty were heavily involved in teaching, and unlike here, weren't doing a lot of research. So as I applied to graduate schools, which seemed to be the best job I could get with a bachelor's degree in psychology at the time, I mean, my grades were good and that kind of thing, but I started to think maybe I need a little more research experience. So I went and did a one-year master's degree in psychology and experimental psychology, and then did my PhD at the University of New Hampshire in physiological psychology. So I got drawn more and more based on my reading and opportunities that came up to doing more of the neuroscience. So my path, I hope this is reassuring to people just starting their academic careers. My path was not one where I knew where I was going from, from birth, or from whatever, kindergarten. It was really one that kind of evolved as it went along based on opportunities that came up and reflection on things that came along. So, and then after graduate school in New Hampshire, I went to a postdoc at the National Institute of Mental Health, and then after 3 years there, I was hired here. So I've been here about 20 years now.
What drew you to applying to Stony Brook to work here?
I'm sorry?
What drew you to applying to Stony Brook?
What interested me in applying
Yes.
here? Oh, well, I applied to about 90 jobs when I was hitting the market. So yes, they had a professor's position in my field, but there was actually something that was a big draw, which at the time they were just putting together a Center for Behavioral Neuroscience, and they had been attracting big faculty from elsewhere, and the psychology department was hiring faculty who were going to be a complement to that center. The center was going to be kind of positioned independently between essentially the more molecular neurobiologists and such on campus, as well as the psychology people who were a little more holistic in their interest in neuroscience. So I seemed to fit very nicely to that, and that was very attractive to me. The other thing, which is actually a reality of life, is that my wife, who was finishing a graduate degree in physical geography at the time at the University of Maryland, also needed a job. So we did what a lot of people call solving the two-body problem, is we both got jobs in our field on Long Island. So she got a job at Brookhaven National Laboratory, where now she's a group leader on a global climate change project, or a group that is part of a global climate change project. So, but at the same time, we were ready to move anywhere, which is often the case with academic types, is you have to go where the job is. So as I say, Stony Brook was definitely a high priority, or at least a top choice, I should say, not a high priority. It was very attractive, but at the same time, I felt very lucky to have gotten the offer back then.
Moving into the research you did, what got you interested in that project?
Oh, the one you've read?
Yes.
Yeah.
Well, okay, so one of the things that happened over, I'll give you the long answer. One of the things that happened over my career here is after, you're probably learning about academics, and we have our phases of life. So our assistant professor years where we're trying to kind of make an independent name for ourselves and get our labs established and learn how to teach and do all the things that, you know, are part of being a college professor. But as I got sort of settled in here in sort of my mid-career, I started collaborating more and more with other researchers on campus, and one of my main collaboration is with Bill Van Nostrand's group over at the Health Sciences Center. And so he's a biochemist who's interested in Alzheimer's disease and has created animal models of, primarily mouse models, where human genes associated with hereditary forms of Alzheimer's are inserted into the mouse genome, and the mice develop pathological symptoms you know, consistent with Alzheimer's patients. So transgenic mouse models are great for doing things like testing interventions, drugs, and that kind of thing, but also studying the time course of the pathology. And of course, with Alzheimer's disease, one of the main pathological, the main set of symptoms that are a problem for people's function is memory loss and other sorts of things, losing language skills, spatial disorientation that get progressively worse. So as you're using these models, you have to both see that the brain changes are occurring that are consistent with the disease, but also the cognitive deficits. So prior to this time, the cognitive deficits that in mouse models, and I came from a rat background, and believe it or not, rats are cognitively much more sophisticated than mice. So, but the field had done the genetics in the mice, and in general, cognitive testing in mice was very simple and focused on more or less one apparatus called the Morris water maze. And this was basically a tub of water submerged in the tub in one of the 4 quadrants of the tub. If you take the big circle and split it into quadrants, one of the quadrants had this little platform, and it was considered a memory test because you plunk the animal into the water and they'd swim around until they found the submerged platform. And then on subsequent trials, tests usually it was on 24 hours between each period, the mouse who had intact memory would find the platform more quickly. So you'd see them swim right to it in the end. But the people had been concerned that too much focus on that particular apparatus, especially since it was a bit stressful for the animals. So the analogy someone said was it would be like taking your grandmother who was showing some signs of mild cognitive impairment and throwing her in the swimming pool and expecting her to find something. It was, you know, that would be kind of the equivalent. That's exaggerated a little, but we wanted to have cognitive testing. We thought we should develop, because this was my background in the operant work, so using automated apparatus and such, that would be more consistent with human neuropsychological testing. So, you know, a battery of tests that are progressively harder, and you see at the point which someone fails, essentially. So that's what we did, was we developed a set of operant tests that most of them were around and tapped different things, but arranged them in an order that had, to my knowledge, was the first time anybody had really ever challenged mice to that limit. And then we looked at both what are called wild-type mice, who are just normal background healthy mice, and one particular classic transgenic strain, one of the earliest ones that was developed as an Alzheimer's model. And you know, even though that strain had been around for years, no one had ever really looked at it thoroughly in that way. So what we noticed was that there were certain things that were impaired in the animals. They were slower at learning certain kinds of things, and other things were spared, or they would learn something, they have initial trouble learning it and would be okay. Well, we also were able to isolate the working memory components, which is what a good neuropsychologist wants to do, is understand, you know, exactly what particular cognitive thing is altered, is not functioning well, and know that it's exactly that, not something else that's affected that looks like that. And again, Morris water maze, the swimming pool, you can, you know, it's just a very crude way of doing it. And then finally, we were able to show that the set of tasks themselves became a kind of how far they could get through the task was itself a measure that correlated with their pathology. So it was like a series of challenges. So we started essentially having animals who would just hit their limit at a certain point, and it was correlated with the degree that their pathology had built up. So as I said, we essentially developed a new tool for the field, and it also allowed us to learn more about this particular mouse strain. Subsequently, we've actually thought of the task, there it functioned as a complex dependent measure in our experiments. We've also been using a similar progressive set of challenges, cognitive challenges, automated challenges for the mice as actually a treatment. So in essence, it's mimicking brain training strategies that there's companies that do this, like Lumosity and such, where people who are concerned about losing cognitive ability will go online and do these brain training games, and it essentially serves as a model of that, we think, as well, and we've been investigating that versus other sorts of lifestyle interventions. So as I said, that was the first article where we demonstrated that you could take these mice for months of training and bring them along under low stress conditions. They actually seem to have, as much as you can tell that mouse is having a good time, they seem to have a good time going to do these tasks and such. So.
You said you've been doing subsequent tests. What's the most recent one?
Well, as I say, we've been doing, one of my graduate students is doing her dissertation comparing the effects of cardiovascular exercise, beneficial effects of cardiovascular exercise on, because a major component of Alzheimer's pathology is vascular. So the misfolded protein that's a major part of the disease, some of it goes and sticks on the capillaries in the brain, and so kind of cleaning that out with regular exercise appears to be, or may be beneficial. There's some literature that suggests that. And then she's using the operant test as a sort of way of isolating the cognitive stimulation without a lot of exercise. So it's a way of getting essentially two beneficial suggestions for people that we can do something about. You can't change your genes, but you can perhaps make recommendations about how much exercise is beneficial, when to start it, how regular it has to be, versus the benefits of the cognitive testing. Both have been shown in epidemiological studies to be, at a correlational level, to be beneficial. So higher level of education puts off the onset of symptoms, and people who are more active cardiovascularly, in better shape, have higher levels of fitness, also seem to have better cognitive functioning as they get into older age. We're not sure what the mechanisms are, what parts of the brain benefit from those, and maybe that they're complementary, and that essentially you have physical exercise and cognitive exercise may, if put together, may be a super package essentially. So again, those are all open questions that we're trying to answer. It will take years, of course. So, but that's what we're doing. So that's kind of the follow up of your article, the article I gave you, which is now 3 or 4 years old.
Sorry. So what was your role, like, kind of specifically in the experiment? Because it's kind of hard to tell just from the paper.
Yes. So the lead author, Alice Blackshear, was my doctoral student, but the Van Nostrand lab and the Robinson lab are heavily intertwined at this point. We have grants together. We've had grants together. So what we essentially have is two PIs, principal investigators, Bill and myself, and then his lab has technicians, graduate students, postdoctoral fellows, and then part of my integrative neuroscience. So my lab has doctoral students like Alice Blackshear and Maria Anderson, and undergraduate RAs, but we tend to go back and forth, even though they're over in health sciences. Most of the histology and the biochemical stuff is done over there. Most of the behavior is done over here. So we're kind of like joined, even though we're in different departments. He's in neurosurgery, I'm in psychology. We're funded by the same sources, and we tend to be a kind of like a two-headed animal in a sense. So my role, the PI role, so it's a big hierarchy, tend to be support and idea generation. We tend to write the grant proposals and spend a lot of time doing that kind of thing, advising the graduate students. The graduate students tend to do the heavy lifting in terms of the work because they have to write their doctoral dissertations, and usually our projects are doctoral dissertations. They're supported by technical staff. There are, again, undergraduates who are often involved in all aspects of the work, actually doing behavioral testing, doing assays. So collectively that becomes the team on the author line. And author lines are always interesting because there is an open question sometimes. Who got first? And why are the two old guys on the end? So in neuroscience, for example, you have what's called senior authors, which are the people who tend to, own the labs, essentially, who run the labs, who generate the money, they tend to go at the back end. It's either because that's where we belong, or depending on, or because it's sort of assumed that that's where you look for the person who's most most senior, versus the person who took primary lead on doing the project, in this case Alice Blackshear, and wrote the first draft of the paper and kind of coordinated all the other people, is usually the first author. Though sometimes, again, complicated collaborative projects, often you'll have to figure out ways of giving, equal credit to the first two people and that kind of thing. So it's not always so straightforward that it's just the graduate student and their advisor. It's these big team kinds of things become. Sometimes papers will have 25 authors, and you never know. Some papers in like physics or whatever, where there's a big experiment on a huge instrument or whatever, might have 100 authors. So because it's very large and expensive and time consuming, and everyone had a hand.
I was going back to the project that you did, you were talking about like possible applications in going from mice to human.
Mm-hmm.
Are most of your projects using mice or rats as a model?
Yes. Yeah, that's my specialty, essentially.
Okay.
Yeah, I'm one of the people here essentially does mouse and rat models of things. Yeah, so and really with the emphasis primarily being on behavior, which is why I'm in a psychology department. I mean, I can do all sorts of other bench techniques and such, but the specialty of my group now is really to do this sophisticated behavioral testing, particularly the cognitive testing. But really, I do everything. We test for, partially because other things can be confounds with cognitive things. So if they're not motivated, they're feeling lethargic, then they're not willing, they're mice, they can't talk to them. I mean, you can talk to them all you want, but they don't talk back. So you have to do various things, and you make inferences that, okay, they're highly motivated to let's say, get the reward in the task, but they can't remember, so that's why they're making mistakes. But if they're not motivated, they're not hungry or thirsty or feeling lethargic, then they may be making mistakes because of that reason. So that's the kind of subtlety of what we try to do, is you test in other things to be able to extract or rule out various other explanations for why there's something wrong with the mouse.
What drew you towards working with rats and mice? Was it something in the past, or is it something that's been done already?
Yeah, well, I mean, neuroscience has a long history with rats and mice, essentially. Primarily rats, but again, with the genetics of the genetic models started to really come about in the early 90s, and then they became a kind of equal partner in this. So, I mean, there's been a very long history. So in all sorts of biology, where you're trying to translate something to humans, so drug development, for example, you always have a phase where it goes from, let's say, pure chemistry and what are called in vitro, or various assays that are on the bench or simple apparatus, to a kind of intermediate step where something is tested out in a rodent model, sometimes in other animal models, but not so much these days, and then you eventually move to clinical trials where you move into humans. But again, basic research has the same complementarity. The assumption is that with the animal models you can control conditions precisely, but you then have to have this imaginative leap when you start translating to a human, and that's again where that's where I come in, is because I am situated among human clinicians who study depression and memory problems and such, as well as basic researchers who are studying cognition and cognitive processes. So the leading research in the world on attention is around the hallway. It's very convenient to be able to go in and say, so we have a mouse task that we think is looking at divided attention. What do you think? They can sit on my students' doctoral committee. So we try to make sure that there's at least a strong analogy between what we're doing with the mouse or the rat and the human equivalent. Therefore, it gives us the validity, the assumption that what we learn in that model system actually is a model. So it's no good to have something, oh yeah, this intervention worked in the mouse, and then not have it work in the person.
People?
Yeah, yeah. So.
So most most of your studies tend to focus on like brain functioning and dysfunction, like using mice as models. Like how, and you mentioned in your paper using like that specific genetic model of that mice. Like how often do you use like different genetic strains of mice and things? Is that like typically, or like how do you do that with the mice basically? How do you like splice in those genes, or is
Oh,
that?
yes, right. Okay, so you have two great questions in there. The first one is, so the molecular biologists handle the creation of the models, and most of the models are standard. So like the 2576 model that was used in that paper was one of the first created in the mid-90s using a kind of older method. But in short, there's several different ways to do it, but basically what you're doing is you have to design a sort of section of DNA with usually a promoter region that kind of turns it on and off. And then you figure out a way to get it, so used to some of the early methods used stem cells. Sometimes you just did what was called microinjecting it into a fertilized egg, and you hoped it found its way into the genetic material of the mouse. You know, you develop several lines, and then you characterize them and screen them, and then you find which one seemed to be the best version of something. So by the time I get them, there's usually, I mean, there's hundreds, anything. There's mice that have had genes removed, there's genes put in, so the ones we're looking at generally are the ones where they've had genes introduced. But usually you'll have these sort of stable lines that have been around, they're well characterized. And so the second part of your question, or actually it was the first part, but the second part is usually what we try to do, so no mouse model is a model of the disease. So they're usually a model of aspects of the disease, and that's why it's beneficial in studies to compare 2 or 3 that have different, you know, sometimes it's different combinations of genes, sometimes it's, you know, where the same genes but under different conditions may produce different time courses of the pathology. So by testing multiple lines, you develop what's called convergent validity. So you know if it's true in one and you see it again in another and again in another, you start to really trust it. So you know, it's not in every paper that you'll see multiple lines, but we try to almost always compare a line. In our case, we almost always emphasize one particular line that we have that was created by our Bill Van Nostrand's group 10 years ago or more, that is where most of the pathology develops around the vascular system of the brain. And then some of the other ones, like the 2576 and the 5X FAD, which tends to develop lots of plaques of this A-beta and amyloid throughout the brain. So those are kind of, their pathologies are very distinct, and therefore they're good, you know, to look at side by side, essentially.
Okay. What advice would you give to like any undergraduate students, like just about doing research or just in generally with school?
Oh, okay, so again, 2 great questions. Yeah. For research, I guess really especially you know, if you're thinking of going into a career in research or in medicine or a medical field, research is great to get at least some of. Not everyone to getting into a lab. I mean, there's only a certain number of labs here. Not every undergraduate can be. You know, labs have to actually do research, so that's their primary focus. But if you can get into a lab, it can be a great experience. It's not mandatory, but really, you know, you see real science in action and you absorb a lot by just being there. So that's a really good thing. Obviously, what I would say that goes with that is to do it passionately. So do it because you're really, really interested in it, not because you're going through the motions. That's sort of the best piece of advice I can give you, is do whatever you're doing be fully engaged. So you absorb the most, you put the most in, you'll have the most fun doing that. So if you really don't want to do research, don't go through halfway. There's other kinds of things that might be, get you much more interested, much more excited, essentially. In general, boy, I could go on for hours on that, but I mean, number one, keep your grades up. That's always, and why it is, you've heard my story. I mean, the one thing, I didn't decide what I wanted to do, but I always did my classes as fully as I can. It didn't mean that I didn't have a bad class here and there. I always like to tell the story. I ended up teaching research methods and statistics here, and the first time I took stats as a freshman, I got a D-, which was a gift. You know, it's just one of those semesters I got, you know, I looked the other way, and next thing you know, that wasn't a bad experience for me, because I you know, like, blah, I guess I got to concentrate on that. But, and, you know, but in general, I kept my grades up. That's essential. But yeah, other than that, as I say, the main two things I suggest is really be immersed and mindful of what you're doing, and do the things that are exciting, that you're passionate about. It tends to work out. That's what I find. I find too many overly stressed students. And you know, obviously there's good reasons to be stressed, but you know, it's, you know, in general, this is a great time. Being an undergraduate is, you know, you don't get to do it again. And a lot of people who are like, you know, have been, even as professors, I never left this environment, but I always think, oh, I'd really like to take some of my colleagues' classes. Well, I did not feel that way always when I was an undergraduate, right? But you know, you look back and say, this is a really unique environment. You know, when you go out, you never really come back. I'm fortunate. I have a great job, and I can kind of live here for the rest of my life, hopefully. But you know, so but recognizing that when you're there is tough, and so to some extent it's you know, by just sort of following the things you're really excited about doing, the courses that you really love. You know, obviously you have to also be grounded in some practicality. So if all you want to do is, you know, art and, you know, this and that and the other thing, and you're not thinking at all, okay, what's beyond this? That's you know, you want to think some, obviously. Another good thing, by the way, is keep talking to people like me. You know, start the habit and go to office hours. That's one of the underused resources that here at Stony Brook. It's professor office hours, and professors don't know everything about everything, which is one of the value of talking to professors, is you realize we're specialists. But we are available to you. It's your time, and we're much more happy to help you with making life choices and, you know, dealing with setbacks and all that kind of thing, than having, you know, you just come in, you know, to talk about grades and those kind of things. That's the stuff we will talk about. We'd much rather talk about, you know, where you're going and those kind of things. So that's, I guess, my third little piece of advice.
Thank you for your time.
Absolutely, my pleasure.
Thank you so much for taking the time to do this with us.
Really enlightening interview.
Yeah.
Great.